Thank you for that lovely introduction. And I want to thank Allison for inviting me to speak to you today. As mentioned, I'll be talking about a new program at the Simons Foundation aimed at understanding the biological bases for sex differences in autism spectrum disorder. Before I begin, I just want to clarify that when I use the term sex, I'm talking about biological, genetic, and physiological characteristics, whereas the word gender focuses on socially constructed characteristics. So you might ask why, from a biological or a medical standpoint, do we care about sex differences? Many traits, in fact, exhibit differences in prevalence in males and females. For example, left-handedness is more common in males. On the other hand, many autoimmune disorders are more common in females. So you can see here in this graph of prevalence with females in orange and males in blue that many autoimmune disorders, including lupus and multiple sclerosis, are more commonly found in females than in males. Many neurodegenerative and neuropsychiatric conditions also show difference in prevalence across the sexes in both directions. So whereas eating disorders and anxiety are more common in females, substance abuse, Parkinson's disease, autism, and attention deficit disorder are more common in males. Some of these differences could be due to environmental factors like differences in diet or employment, but many of them also have underlying biological reasons. And better understanding of these sex differences can lead to better diagnosis or early detection of conditions, as well as more effective treatment. So if we take stroke as an example, in a kind of stroke called ischemic stroke, early in life, it's actually more common in males, but around middle age, rates start to increase in women. Studies in animals have shown us that the hormone estrogen is protective against stroke. And so it seems that as estrogen levels decrease following menopause, the incidence of stroke increases in women. In addition, aspirin, which is given as a preventative treatment for stroke, is actually more effective in women than in men. So understanding these kinds of sex differences can lead to better diagnosis and better intervention for a number of conditions. So as I mentioned previously, and as mentioned in the previous talk, there is an observed higher prevalence of autism in males than in females. And depending on the population and depending on the year, as we learned in the previous talk, that difference may vary somewhat, but on average, it's about 4 times as many men as women are diagnosed with autism. As also nicely mentioned in the previous talk and in the Q&A, which was a really nice setup, there are differences in presentation of autism between males and females. So whereas males may show more language impairments, females may show more motor impairments, and these differences can affect diagnosis. Social and cultural factors can also affect diagnosis of autism in males and females, as can differences in the existence of co-occurring conditions. However, despite these differences in diagnosis and presentation, there is likely a real underlying biological difference in susceptibility between men and women. And while there are some hypotheses, the underlying biological reasons or mechanisms for this difference in prevalence is not well understood. So while we don't yet understand the biology of sex differences in autism, we do actually understand quite a bit about mammalian sexual differentiation. Sex is initially determined in mammals by genetics. So as you know, DNA is packaged into chromosomes, of which humans have 23 pairs, 1 set from mom and 1 set from dad. One of these pairs, designated by the letters X and Y, determines sex. So the presence of a Y chromosome results in a male, and the absence of a Y chromosome, which usually means 2 X chromosomes, results in a female. Early in development, the presence of a Y chromosome, and specifically this one gene on the Y chromosome, SRY, determines whether an animal will develop testes or ovaries. These ovaries and testes then release estrogen or testosterone, which we refer to as sex hormones. And those sex hormones set up different patterns of anatomical development for the long term, as well as stimulating certain behaviors in a more short-term way. The presence or absence of androgens like testosterone also during prenatal development also lead to sex differential development of the brain. And I just want to emphasize that these actions of sex hormones are happening really early in development. So I know most of us probably think about hormones in the context of puberty, but this is happening really early in embryonic and early postnatal development. Disentangling the effects of genetics and sex hormones can be really difficult in humans, but in animal models, this is something that we can do. So in thinking about what the underlying biological reasons for sex differences in autism might be, there are many potential hypotheses. I'm going to discuss 3 potential hypotheses today. The first is that sex chromosome genetics underlie sex differences in autism. The second is that differences in sex hormones underlie sex differences in autism. And the third is that differences in a specific tissue or cell type underlie sex differences in autism. And I want to emphasize that these 3 hypotheses are not mutually exclusive, so they could all be true. So the first possibility is that sex chromosome genetics underlie sex differences in autism. So as I mentioned previously, females usually have 2 X chromosomes, whereas males have an X and a Y. And there's this one gene on the Y chromosome called SRY that determines whether testes or ovaries develop. But there are other genes on the Y chromosome as well. Most of these genes on the Y chromosome have counterparts on the X chromosome, but there are subtle differences between the versions of these genes on the X chromosome and the version on the Y chromosome. So these differences could lead to sex differences in autism. In females, which have 2 X chromosomes, only one of the X chromosomes is usually expressed or turned on, and the other one is suppressed. And this happens in a random process in each cell called X inactivation. And what this means is that females are essentially a mix of cells expressing one or the other X chromosome. As a result, if you have a mutation on the X chromosome, usually the effect is less pronounced in females because we are this mix of cells expressing the 2 X chromosomes. So the intact X chromosome can sort of dilute the effects of or compensate for the effects of a mutation on the other X chromosome. So many conditions that result from mutations on the X chromosome, like for example, Fragile X syndrome, are more severe in males than in females for this reason. On the other hand, some mutations on the X chromosome are so severe that males with their only 1 mutated X chromosome, the embryos don't even survive. And so you only end up with females who have a condition as a result. And this is, for example, the case with Rett syndrome. So either of these mechanisms could also be contributing to the sex difference in autism prevalence. The second hypothesis is that sex hormones like estrogen and testosterone are contributing to sex differences in autism. As I mentioned previously, sex hormones set up different patterns of anatomical development over the long term, as well as stimulating certain behaviors in a shorter term way. And the presence or absence of androgens like testosterone during prenatal development lead to sex differential development of the brain. In the brain and in other tissues, hormones act by entering cells from the bloodstream. So hormones are circulating in the bloodstream. They enter a cell where they bind to their receptors. Their receptors then travel into the cell's nucleus where they directly regulate gene expression. So depending on whether estrogen or testosterone is present, you have different sets of genes that get turned on. And these can obviously lead to differences in cell and circuit function. So these differences could be underlying the difference in prevalence of autism between males and females. The other 2 hypotheses that I discussed are fairly broad, and they were agnostic about where sex chromosome genetics or sex hormones might be acting to affect the prevalence of autism. It is also possible that genes or hormones could be acting in a specific tissue or cell type, and that's what this third hypothesis is. So there are multiple cell types in the brain. There are the neurons that get all the attention, but there are also a group of other cell types that are collectively referred to as glia, and they play really important roles in supporting and regulating neuronal function. In particular, one type of glia called microglia are the immune cells of the brain, but they also act to regulate the development and the plasticity of neural circuits. Microglia have been shown to have differences in their gene expression and their function between males and females. And independently, microglia have also been implicated in the etiology of autism. So it could be that sex differences in microglia are underlying sex differences in autism prevalence. Now, as I mentioned previously, these three hypotheses are not mutually exclusive. And particularly here, if there are sex differences in microglia, they are driven either by sex hormones or by sex chromosome genetics or by both. So we have these three hypotheses, but in order to test these hypotheses and additional hypotheses, the Simons Foundation initiated a program called the SFARI Collaboration on Sex Differences in Autism. We held a request for applications in 2020. And the goal was to examine sex differences in brain function to identify key differences in gene, cell, and circuit function that account for the observed differential sensitivities of the two sexes to autism. As a result of this RFA, we funded 5 projects totaling $28 million in funding over 4 years. Each project is made up of a multidisciplinary team of principal investigators. Overall, there are over 20 principal investigators and institutions involved in this program all across the world. The 3 hypotheses that I discussed on the previous slides will be explored in these projects, as well as some additional hypotheses. The project started last summer and early this year, and we expect they'll go for at least the next 4 years. We really see this as a collaboration of collaboration. So even though each team is made up of several labs already, we expect the 5 groups to really work together and collaborate across the 5 teams. And we're working with them to set up a robust data sharing infrastructure, both within the collaboration and with the broader research community. We expect that these projects will generate valuable resources that will help to answer other questions about autism, in addition to investigating the difference in prevalence and presentation of autism across sexes. And if successful, we think these projects could lead to clinical advancements like earlier diagnosis, since females are diagnosed later on average, better understanding of the heterogeneity of autism, perhaps the identification of subtypes of autism within or across sexes, and the potential for interventions based on a better understanding of the biology underlying sex differences in autism. Thank you for your attention. I'm happy to take any questions.
If you have a question, just raise your hand. Question over here.
Thank you. Was there any thought about studying postnatal medical records or special environmental exposures, like the first 2 years?
I do believe one of the projects will be looking at, I don't know if they'll be using like electronic medical records, but they will be looking at, clinical data from a cohort of people, I think, into early life. The project is not directly focused on environmental factors, given that, we're really trying to disentangle the underlying biology of sex and how that interacts with autism.
But are they, anyways, it's a very rich source of information the first 2 years, medical records. And given the fact that about 40% of people with autism have significant co-occurring conditions that begin in this period. Anyways, it's something to think about.
Other questions? Question here.
Sorry, I'm a little blind up here. The lights are really bright.
That was a really great talk. I'm really curious, given what we know about gender and also sex in autism, how sort of these gender issues come into play, sort of come into conversation with sex differences work. I know that people approach it really differently, but if there are people who push back on sex differences research for some of those reasons. And I'm just curious how those kinds of additional analyses may come into play, or what kinds of information or signal you might be getting there.
Yeah, so I think, I said that we know a lot about sex differentiation. I think we don't know anywhere near as much about, how gender identity develops. And also given that that tends to happen a little bit later in life. I think this project is probably realistically not super likely to shed a lot of light on that. Also, a lot of the work is, this is really basic science research. And so a lot of the work is being done in experimental models or looking at human genetics. So I think we're best placed to answer questions about sex rather than gender, but I do think that's a really interesting area and an area definitely ripe for future research.
Hi, you had a great talk. Thank you. Sorry, I wish I had more of a voice right now. So I'm from the Seaver Center, and so the lab focuses on DDX3X syndrome, which for those of you who may not be familiar, is a genetic mutation more present in girls than boys and affects autism presentation. And I'm just wondering if any of the projects funded are looking at the genomic basis for something like DDX3X syndrome. Thank you.
Yes, definitely. So multiple of the projects are looking for differences in the distribution of different genetic variants between males and females. And definitely DDX3X is one that's already, some of them are on the X chromosome, and so they're sort of more obvious, but definitely that's one that's already come up. Yeah, thanks.
Hi, thank you so much for this talk. Could you elaborate on the differences that the microglia have within the sexes and whether or not that would impact plasticity and how that could change with time?
Sure. I'm just going to go back. Oh, I don't know if I can go back. Yes, I can. I'm just going to go back to that slide. It's probably pretty small, but yeah, so like different functions of microglia have been shown to be different across sexes. And so, for example, you can see that different inflammatory related genes are expressed differently across the sexes. I think these definitely can have impacts on plasticity. I don't know how far along the research is in terms of following specifically which of these differences are most important for plasticity. But for example, the increased phagocytic capacity in female microglia. So microglia are known to phagocytose synapses, and then that obviously regulates plasticity during development and later. So I think definitely some of these changes are likely to impact plasticity, and that would be how you would imagine you would get longer term changes in brain function as a result of sex differences in microglia.
Thank you again.